One difference between myself and the previous presenters is that I'm not a medical doctor. I'm a PhD, even though after my talk I've been accused of being a medical doctor. Another thing that is continuum is that both Graham and Dr. Owen grew up in Sault Ste. Marie, Ontario. Well, I grew up in Sudbury, Ontario, just down the road. So we are all interconnected in one way or another. So I just wanted to acknowledge in the beginning that the work I'm going to be talking about here is an initiative that the provincial government in Manitoba through Research Manitoba initiated, trying to bring researchers together to have an impact on the healthcare system. And we put a proposal together for CLL, bringing people together in order to have an impact on the CLL patient. And before I start, I'd like to also acknowledge that I have a disclosure. It's the first time ever I've been able to say that I have a disclosure. I do have funding from Janssen for a research project. So let's see here. So the cluster, it is an organization of researchers whose focus is on CLL, but crosses many different fields. We have population-based scientists, clinician scientists, basic researchers, and nurses working in our group. They cover the entire spectrum of CLL research, from the patient and care all the way to the basic mechanisms of action in CLL. And these are just an example of some of the researchers in our group. But really, at the core of what we do is the CLL patient. So everything that we do in our group is we have to ask the question, what is relevant to the CLL patient and how can we have an impact on the CLL patient? And so we have developed projects within the cluster that try to answer that question. Either it's through better patient management, better clinical care, better targets for therapy, or trying to understand the biology of CLL through technology, were all projects that were designed around this. But none of this would be possible if we didn't have a foundation to grow on. And our foundation is that we have a dedicated CLL clinic within Cancer Care Manitoba in which we follow CLL patients. Cancer Care Manitoba is the central cancer agency that serves the province of Manitoba, in which every patient who's diagnosed with cancer will be associated with the cancer center at some point in their journey. And we have a CLL biobank in which we can capture patients' material and connect it to a clinical database in which we track patients over time. To better describe this foundation that we're doing, we have a CLL Tissue Bank in which we have over 1,500 CLL patients that have been kind enough to donate their blood samples to the bank over time. And we bank this over the course of their disease, so we have multiple samples from these patients. We also ask their spouse and family members if they want to donate to our bank as well, because we know that we have to have normal controls for our experiments and to compare it to our patients. As well, we bank patients before and after treatment, as well as banking specialized materials such as plasma or live cells to be used in projects moving forward. I'd just like to say before I forget that we thank the CLL patients for their dedication, for allowing us to have the privilege of storing their samples and having their clinical data to do this research. We track over 2,000 CLL patients throughout their journey, and these patients could include MBL, CLL, SLL. And we track both clinical and biological markers of these patients, what type of treatment they get, and the changes in their rise stage and lymphocyte doubling time through the course of their treatment or journey. The CLL Clinic at Research Manitoba is one of the few dedicated CLL clinics in which all Manitobans who are diagnosed with CLL will have the opportunity to be referred to. And we have an active program in which we tell physicians who have a CLL patient that it is a resource that's available to them. These clinics are led by Drs. Johnson, Dhaliwal, and Banerjee, in which you had her talk earlier today. It runs clinical trials as well as education opportunities for patients throughout the course of their disease. So what do? We have these fantastic resources. How are we going to utilize them? Well, the first part of our goal is to improve management of CLL patients. What do I mean by that? We want to know what type of comorbidities these patients get, what type of infections they get, and second, cancers if they get them. We want to then use this to develop policies of how to better manage these patients with these comorbidities. To do this, we have to have a cohort of patients that we can collect all this data on. So we decided to take a cohort of patients from 2005 all the way to 2014-15 over a 10-year period and track all different treatments, comorbidities, infections, and other clinical parameters of these patients to see how they do. Now, you've seen this slide about the changes in therapy that's happened in CLL over the years. So it is important to note that these patients that we are tracking have mostly received fludarabine, cyclophosphamide, and rituximab therapy through this, and a subcohort would have received chlorambucil as well. This was before ibrutinib and venlafaxine, which occurred in 2015, 2017 to now. That will be part of a future cohort that we will be developing in the near future. So how did these patients do? Well, they did as well as what other studies have shown, which is good because we wanted to know how well we did compared to what other centers did. And so what we found is that of the 632 patients that we tracked, 67% didn't require therapy, while 33% did require therapy of some sort. That is approximately what you would have seen if you were in Europe or in the United States. Of the patients that were untreated, about 13% died during this period of time, while patients who were treated, around 27% died. Next, we went to look at the biomarkers that people have been talking about for these patients, which include their age, gender, comorbidity status, that's SEERS, the lymphocyte doubling time, LDT, their CD38 levels, as well as ZAP70 and the mutational status of their immunoglobulin gene. So the reason why I have the immunoglobulin gene highlighted here was it was the only parameter that both successfully predicted time to first treatment as well as overall survival in our patient population. The other parameters were either good at predicting overall survival or time to first treatment, but not both, suggesting that the immunoglobulin status is an important marker that we would need to continue to evaluate. When we looked at the cause of death of patients in this cohort over the 10-year period, we found that patients who had chemotherapy during this period either died primarily of CLL or infections or progression of CLL such as Richter's. This isn't surprising because patients that need therapy because their disease is aggressive. And so if the therapy wasn't successful, they would succumb to the disease. However, when we looked at patients without any chemotherapy, what we were surprised is there was a significant amount of patients who died of second malignancies. When we tracked the cohort in terms of the incidence of second malignancies to the general population, what we found was there was an over 2% increase in the risk of getting second malignancies than it was in the normal population, even when you compare it by age. This was also observed by other groups in other countries as well. What we wanted to find out was, well, what is the factors that could be involved in patients getting second malignancies? So first of all, we looked at the incidence of skin cancer in our population, both before and after diagnosis, to see is there any stage within the disease that would indicate they would get skin cancer. And what we found is that patients could get skin cancer at any point during the journey of disease, even before they were diagnosed. However, it seems to peak after diagnosis within the first 10 years, and then after it declines. And so there is something we believe that is associated with CLL that is allowing other cancers like skin cancer to progress in these patients. When we looked at patients who got treated and not, and looked at skin cancer, what we found was that patients who were treated seemed to have a higher level of skin cancer as compared to patients who weren't treated. Also, if you had a solid tumor, that didn't seem to make a difference in terms of the incidence between treatment and untreated patients. Again, suggesting that there is something going on between the CLL cells and the skin cancer within these patients. So then we went back to our prognostic markers and asked the question, can any of this predict the second malignancies within this population? And what we found, interestingly enough, was that both gender and age were good predictors of secondary cancer, which again were not terribly surprising because these were same predictors you would find in the general population. However, the immunoglobulin status was a good predictor, as well as CD38 and SLL. Treatment also came up as a predictor. As you saw from my other side, there was an increased incidence of skin cancer after patients have gotten treatment. What was interesting is that when we put all these parameters together, the only two things that came up as strong predictors was gender and age, suggesting that this is a multifactorial issue within CLL in which we have to further investigate some of the biology behind this to understand why these patients are getting getting skin cancer. One of the reasons that came up, and it's been mentioned by other speakers, and especially Dr. Tips, is that there's an interaction between CLL and its microenvironment. In other words, the other cells around CLL. And one of these interactions is with the immune system. CLL cells influence normal B cells and T cells. And they do this through cell-cell contact, through production of proteins we call cytokines that influences the function of the immune system. And if you're treated with antibody-based therapy, and even with ibrutinib and vincristine, there is effects directly on normal T cells and B cells within a patient. So we wanted to ask the question within the patients that we see, can CLL change the nature of the immune system? And then from there, can we further make some questions as to why this is associated with skin cancer or not? This is ongoing research. I'm not going to explain this graph in any detail. What I want you just to understand is that there is an increase in T cells within CLL patients of the CD4 class. This is a type of T cell helper, and the reason why it's important that it's upregulated is that it's not CD8 positive T cells. These are the cytotoxic T cells that would attack cancer. And the type of CD4 positive T cell that's in CLL patients seems to change over time, and the percentage of these different subtypes seems to be altered in CLL cells compared to normal cohorts. Also, when we looked at patients treated with ibrutinib, these subsets of T cells go down over time. But one thing to note is that they don't all go down. Some go up. So there's this change in T cell subset that's happening. So we are continuing to monitor these patients and looking at the changes in T cell, and then hopefully in the future be able to correlate this to things like second cancers. So what does this mean for the CLL patient? Well, what it means is that we have a dermatologist now that's associated with the CLL clinic that will be referred to by physicians in the clinic to be monitored on an ongoing basis for second malignancies. We counsel patients to be screened for second malignancies, as we know that they have an increased risk. We advise patients on prevention strategies, mostly preventing direct exposure to sunlight as an example. And we have education of newly diagnosed patients who are not part of the clinic, that could be out in the community, we give them an invitation to come to a seminar in which we go over the risk factors for them so that they can be educated in terms of protecting themselves in the future. So in the future, what we need to understand is what is happening to the immune system in CLL patients. How is this affecting the CLL biology? Is this direct or indirect? We need to analyze the type of infections these CLL patients get, and we need to look at what role the microenvironment plays in these interactions. And this is part of ongoing research within our team. The next goal is to improve clinical care. And this project is spearheaded by Aaron Strew and Dr. James Johnson. And this is, again, associated with the immune system in that it has been observed that patients get increased infections over the course of their disease. And one of the therapies that many patients get is immunoglobulin replacement. And what happens is that within our center, they would come and get an injection within the chemo room of immunoglobulins, and they would have to sit in a chemotherapy chair for hours on end and then go home. We thought that there might be a better way of doing this. And so we instituted a program for the subcutaneous injection of immunoglobulin within patients on an outpatient basis. This is a graph of what a patient looks like over the course of their disease, in which they have elevated levels of immunoglobulin gene and could have higher levels of other types of immunoglobulin genes, such as immunoglobulin gene A, immunoglobulin G, and M. And when their disease progresses, what happens is that the immunoglobulin genes drop. And they drop to a level in which they are more susceptible to infections. And when they get infections such as pneumonia, we replace the immunoglobulin in the clinic. So what we do is we train the patients on, and this is Aaron Strew's job, to inject the immunoglobulins through their stomach area by just using a needle and a syringe, and they just push through force the immunoglobulins into their subcutaneous space. And so this is a much smaller volume than they would get in the cancer center, and they could do this repeatedly over time so that they could maintain their immunoglobulin level. Why is this important to a patient? Well, one benefit is it's portable and it's convenient. You can travel. You can go to Florida in the winter and have this. It's important to a lot of patients, actually, in Manitoba. But more importantly is that they can do it in the convenience of their own home. They don't have to go to the cancer center, which can be very stressful to a patient. But more importantly, does this work? So we tracked 90 patients with this injection technique, and we looked at the quality of life, the rate of infections, the visits to the cancer center, and the impact on costs, even though you guys might not care for costs in Canada because it's borne by the French government, but my administrators want to know what the costs are. So what we found, and I'm going to be very brief in this, is that 80% of the people who got these subcutaneous injections were very satisfied with the process, and their overall quality of life increased. And even though it's never in the excellent range because they still have CLL, we can say that their quality of life has gotten from the low good to the high good. Where use of antibiotics was reduced when they were on this subcutaneous injection, which was great, and he's saying that it is working and reducing the infections in these patients. And the cost savings. Because we were injecting lower volumes of immunoglobulin and because they were not coming to the cancer center, we saved on drugs, we saved on nursing time, and we freed up the chemo chairs in order for other patients to have advantages for it. So in other words, we almost recovered the cost of our granting program based on this one project alone. So going back to this patient, so we know that we would need it based on the symptoms of a patient getting immunoglobulin therapy. is there a way we can go back to this cohort and predict which patients are going to need this therapy in the future, or what patients are going to progress based on their immunoglobulin levels? And so what we looked at was comparing the immunoglobulin levels of G, A, and M. These are different types of immunoglobulins, and we wanted to see if this could predict time to first treatment and overall survival. And what we found, which was very interesting, was that only IgA levels predicted time to first treatment as well as overall survival. IgG did not, which was interesting because IgG is the one that really is high and it drops when you need immunoglobulin therapy. So there's something going on with this particular immunoglobulin. In data that I didn't present here, as I just got it recently, is that it also predicts for immunoglobulin replacement therapy as well. And an internal control is our mutational status. If you are unmutated, you are, again, of a higher risk of getting immunoglobulin replacement therapy as well as time to first treatment and overall survival. So what this means is that we now have a way to decrease the infections of patients and increase their quality of life without them coming into the cancer center. This is going to be implemented as our standard of care this year. And through the monitoring of immunoglobulin levels, we could provide evidence for aggressive disease and potentially increased infections as a monitor over time. So we're going to continue to monitor cost and quality of life of these patients moving forward, and we're going to correlate the IgG levels with infections and the replacement of immunoglobulin genes to see if there is any trends that we can come up with. Tomorrow, Erin Strew is going to give a talk about self-care and secondary cancers, and she'll go into more details about what I've described here today. The second goal is to create investigator-initiated clinical trials. Can we have better therapies for CLL patients, or at least a rationale for future clinical trials? This was work done by Dr. Johnson and myself. So we went through a whole series of drugs and we looked at the synergy these drugs have. And this means that the combination of these drugs together would give more killing to CLL cells than each individual drug by themselves and or together. So it's more than additive, so that it is doing something that is better. And the reason why we wanted synergy as our final endpoint was that we wanted a rationale to reduce the concentration of these drugs because these drugs are toxic. And so you don't really have to understand how we did the experiments. All you have to understand here is blue is good, red is bad. And so this combination of bendamustine and idaalisib was the best combination that we found in the lab to induce killing in CLL cells. And we're now doing an ongoing projects to find out why. Why is this there? Because if we understand the mechanism, then we can probably better understand how to rationally use drugs in combination moving forward. To further the idea that we want to reduce toxicity in CLL, we looked at repurposing FDA-approved drugs to say, are these going to be effective at inducing cell death selectively in CLL cells? And so we used ceramacine as a antidepressant drug that was in clinical trials and their family members that were FDA approved. And we showed that we're able to kill CLL cells with a mutation in P53, as well as not killing a patient's T cells. And I can also say that they don't kill normal B cells as well. We also used antihistamines in combination and alone, and this graph just shows you that we were able to kill CLL cells to a greater extent than normal cells at lower doses, which is what we call a therapeutic window. That's the separation between these two curves. And when we used it in combination with ibrutinib, we saw increased killing. So this suggests then that we could use drugs like antihistamines in combination with ibrutinib as maybe a way of, again, trying to reduce toxicities. Before all of you go out and buy antihistamines off the counter, I have to tell you something. The concentrations we're using are nothing that you can actually get by looking at these drugs off the counter. So probably will not work. That said, what we need to do is we need to test this in an animal model. We need to find out what doses are going to be effective in combination with other drugs such as ibrutinib. And then we will take it to phase 2 clinical trial to see if this is going to be effective in CLL patients. So that will be the future. The final 2 aims are more about discovery. So we wanted to determine, can we find novel targets for CLL? And one of the areas that we looked at was metabolism. And so this is work that was done by Dr. Banerjee, and she was interested in understanding the biology of CLL, because if you don't understand the underlying biology, we will never be able to have effective therapies that will kill CLL patients and not kill other cells. And this is her technician in her lab with her. So why metabolism? Well, metabolism is altered in cancer. It's one of the hallmarks of cancer, actually. And it provides a survival advantage to cancer cells, and it's been shown that in CLL there is an alteration in metabolism. So can this be an attractive therapy for CLL? I'm not going to spend a lot of time on this graph. This graph is basically showing you that we can measure metabolism in terms of the oxygen consumption within a cell, and that CLL cells have higher metabolism than normal cells or less aggressive CLL. When we use a metabolic toxin, we can be able to kill CLL cells again at a higher rate than we would if you're looking at normal cells that are taken from a healthy individual. So again, thinking that we have a therapeutic window, we are now looking at the mechanism of action of these drugs and how to use these drugs in combination to go into animal models and eventually to clinical trials. So we need to further understand then the changes that occur in metabolism and target those cells that have these changes to better effectively treat CLL. We will do this through the use of animal models and eventually be able to conduct phase 2 clinical trials in the future. The final thing that I'm going to talk about is PI3 kinase. This is a target for idelalisib. It is one of the drugs that has been shown to be effective, but one of its greatest problems is its toxicities. So we wanted to get down to the biology. Why does this drug work, and can we make these therapies more effective? This drug works because it takes the CLL cells out of their microenvironment, as Dr. Kipps described. The microenvironment is really what causes the problems with CLL. It gets into your lymph nodes, it gets into your bone marrow, and screws things up. And it is the stromal cells and the interaction with CLL cells that provides the fuel, the survival, and the side effects of CLL in patients. And what we wanted to do is see what role PI3 kinase plays in this, and can we make better effective therapy. So we want to deny the CLL cells to their microenvironment, and we want to increase their susceptibility to therapy. This is just a picture showing CLL cells in collagen. This is the assay we use to look at the movement of CLL, because the movement of CLL will determine how it gets into the microenvironment. And we wanted to know, using PI3 kinase inhibitors, what will happen. What we found is that if we target more than one type of PI3 kinase inhibitor, we can block migration of these cells. And migration is the first step of these cells getting into the microenvironment. And we found that inhibiting one type of isoform of CLL in combination with another gave effective therapy. So we found that the PI3 kinase gamma, so this is not the same type that ibrutinib is associated with. I'm sorry, not ibrutinib, but idelalisib. So that's the PI3 kinase delta. That's what it's targeting. We found that gamma is upregulated in CLL. We found that if we put in this isoform, we can increase migration. So this gives a rationale that we need to target multiple isoforms of PI3 kinase if we wanted effective therapy. So we are continuing these experiments to see what effect this has on both migration and adhesion of CLL cells to other cell types. We need to do this in animal models. We need to create microenvironments for this. And furthermore, we have to see how these drugs affect the microenvironment. What effect does this have on T cell function or on stromal cell function in the microenvironment, and how these things can interact with each other? Finally, all this work would not be possible without collaborations from the CLL community. As has been mentioned before, we have a national CLL meeting in Winnipeg in which we bring in people from across Canada and the United States and occasionally Europe. And from this, we have developed collaborations within multiple centers and connected people together. For example, Chris Oakes at the University of Ohio actually was in Europe as a postdoc that first came to our meeting. He met people at our meeting that created collaborations and also eventually led them to Ohio State University and create networks of collaboration. So these are the things that we want to promote. It's not just us, it's the entire community, because at the end of the day, it's about the CLL patient. So thank you for your time, and I can take a few questions.
Thanks so much, Spencer. That was really a great update of the fantastic work that you and your colleagues are moving forward in Winnipeg with. We do have a few minutes before the next speaker for questions. I think I see a hand in the corner over here.
Now, I seem to recall sending via FedEx a sample of my blood to Manitoba. This would have been about 8 years ago, possibly.
Yeah, I remember.
Thank you. You got it then. But what I have noticed here in Ontario is that despite an extensive database called eHealth, privacy concerns seem to be the default. So I have to insist that Dr. X and Dr. Y know exactly what Dr. C said. If I just leave it to them to do it, they won't do it. Can I assume that you're still getting updates from Dr. Buckstein at Sunnybrook about my status to go with the study that you're doing? Or do I have to sign something else?
It's a good question, and it is about healthcare databases and access. And it's actually a problem in Manitoba in that we have trouble accessing patient health records because even though it's electronic, I put quotes on it, it's not all electronic. Somebody has to physically go in there and take the data out and bring it to us. So we haven't pushed it. We got your initial clinical data, but we haven't done the follow-ups because we know that this is a real issue within the healthcare system. And we're trying to automate what we're doing in Manitoba as well. So to answer your question, no, we haven't, but we're working towards a system in which we will be able to effectively do that. But at the same time, we will be able to use your sample because we have the baseline data.
Okay, so I don't have to do anything more. Thank you.
Dr. Gibson, pleasure to be here and have your presentation. I was very intrigued with a patient power interview that Andrew did with you back in, I believe it was 2012, in which you were investigating potential therapy targeting ZAP70. And part of being in an educational group like this and the advantage of being a proactive patient is to gain that, what I call the casino's edge on telling what therapies might be giving you a little better advantage over another therapy. And I was wondering if many patients know that ZAP70, because it's focused in CLL, is so critical in the immune function on T cells. And I never heard of anything that came out of your studies. And I was curious to know if you could give us a little heads up on what types of insurance, when you're targeting something like ZAP70, even theoretically, do you have to be aware of, since you can't really try it on humans, and how effective it is in deciding whether or not it might be a danger, for instance, if you're inhibiting ZAP70 and it gets inhibited on the cells that it's meant to function on?
Yes, and one of the other issues with gefitinib is that it also targets the EGF receptor, and that causes other types of side effects as well beyond the T cell issue. So there is a concern. I think that study demonstrated that if we could target ZAP70 kinase activity with a effective specific inhibitor, that it would be clinically effective, and there's people that are developing SCIG inhibitors clinically that might be better than gefitinib in this regard. But we did publish that work.
Does this have anything to do with conjugating it with other known cofactors like CD27 and I believe it's another PKM2 enzyme or protein that's a proliferation?
Yeah, so you're talking about combination therapy there, right? We have done some studies on that, and it didn't seem to be very effective in our studies, but I think it needs further research.